No, no, per la prima fase.
Da dove inizia? Da qua? Dobbiamo iniziare. Filmate dopo. Ok? Grazie. Sì, no, ma è meglio. Da dove inizia? Da qui?
Sì, 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 sì. Mettiamo qua, ci mettiamo e Danilo passò al Caivano. Vabbè, ora vieni.
Non siete pronti? Noi siamo già online.
Se l'unica cosa che devo fare acceso. Si sente? No. Ok, buongiorno a tutti, grazie per essere intervenuti. E come avrete visto è un evento relativo ad un progetto Horizon Europe Seeds, che è una bellissima iniziativa della nostra istituzione, dell'Università degli Studi di Bari Aldo Moro, con l'intento molto importante di creare il seme dal punto di vista progettuale per poter giungere a progetti europei. Un altro aspetto molto forte di questi progetti è la contaminazione, termine usato dal nostro magnifico rettore, nel senso di mettere insieme in maniera molto trasversale competenze che sono presenti qui nella nostra istituzione per poter esprimere le idee progettuali nella maniera, se vogliamo, più larga e più wide possibile. E per quanto ci riguarda, noi in questa prima fase di introduzione, io dirò molto brevemente il senso del progetto Sepsis, che è il progetto H-Seeds che stiamo portando avanti con i colleghi. Vi parlerò anche della composizione di questo consorzio e c'è già stato un passo molto importante relativamente allo sviluppo dell'idea progettuale, passando da una scala nazionale, direi locale, ad una scala europea, con un progetto che è già stato scritto e sottomesso, e quindi siamo in attesa a livello europeo nel contesto dei bandi Horizon Europe 2021-2027, e quindi c'è già la speranza che possa di fatti diventare anche un progetto europeo. Non a caso fra i relatori abbiamo il dottor Michael Rutzler, che è di fatti il PI di una delle unità di questo progetto europeo. Secondo solo. Non entrerò nel merito degli aim, degli scopi del progetto Sepsis che stiamo portando avanti, semplicemente perché le relazioni che seguiranno saranno incentrate sul progetto Sepsis, quindi presenteremo già dei risultati in termini di attività scientifica, sperimentale, e quindi è inutile anticiparli già da ora. Come vi dicevo, l'ultimo scopo, come avete modo di vedere fra quelli principali, è proprio last but not least, si direbbe in inglese, è quello di poter portare il progetto a livello europeo. Relativamente al consorzio, il consorzio è costituito, come vedete, da 4 unità, 4 nodi. C'è l'unità bio, che comprende come componenti me e le colleghe, la professoressa Tiziana Sorlazzi, la dottoressa Doron Tolomeo. C'è l'unità med di medicina, di clinici, il professor Vacca, il professor Antonio Solimando, il professor Vacca noto a tutti, il professor Angelo Vacca, e il professor Roberto Ria. Unità di biostatistica, il professor Massimo Bilancia, ne è il PI, il professor Fabio Manca e la professoressa Claudia Marin. E abbiamo una componente importante, la componente dell'unità IUS, coordinata come PI il professor Massimo Di Rienzo, che tutti noi conosciamo, e anche il professor Nicolò Mucciaccia e il professor Chionna e Lobuno. Vi dicevo che dall'idea del progetto Sepsi, e avremo modo di sostanzialmente di capire che alla base di questo progetto c'è la volontà molto forte di mettere insieme delle competenze per poter, con un approccio di intelligenza artificiale, fornire nuovi ed efficaci strumenti, soprattutto dal punto di vista prognostico per questa patologia, della quale i colleghi medici avranno modo di sottolineare l'impatto, la prevalenza di questa malattia e la severità di questa condizione patologica. Quindi stiamo mettendo insieme competenze diverse che avrete modo di vedere per poter il più possibile creare degli algoritmi di intelligenza artificiale che possano aiutare in tempi praticamente reali i clinici nella gestione della patologia, essendo una patologia con una rapidità molto preoccupante e quindi è importante poter in tempi molto rapidi sapere la maniera più efficace per intervenire. Detto questo, darei la parola al professor Angelo Vacca. Il progetto, dimenticavo, ma questo ce lo dirà benissimo il professor Vacca, nasce l'idea dal centro interdipartimentale di telemedicina, il CITEL, del quale mi onoro di far parte grazie all'idea del professor Vacca, che ancora una volta ha messo insieme competenze molto complementari, quindi l'origine di fatti è il CITEL, il centro interdipartimentale. Prego, Angelo.
Grazie Giuseppe per questa introduzione. Io mi limito solo a dire che è un progetto oggi molto importante circa la sanità e le spese in sanità, in quanto che la sepsi è una patologia a parte molto frequente, ma che è molto costosa da seguire e da curare. Infatti coinvolge soggetti anziani e polipatologici e che stanno nelle nostre cliniche addirittura per un mese o un mese più per poter essere curati, e quindi questo vi fa capire quanto sia elevata la spesa e non solo, ma anche la difficoltà nella terapia di questa malattia. E in effetti diciamo, molto spesso questa malattia non recede e porta all'exitus, in quanto che coinvolge soggetti polipatologici con difese immunitarie molto precarie. Ora, la medicina che si profila al nostro orizzonte non è più una vecchia medicina, e anzi vorrei dire che la medicina non è più una questione di soli medici, ma coinvolge, ovvio, numerose professionalità per poter approcciarci oggi al malato con le nuove terapie, ma anche con il nuovo modo di comportamento, di pensiero all'approccio medico, alla diagnosi e alla terapia. È necessario avvalersi di numerose professionalità. Ora mi onoro di essere coordinatore del CITEL, che raccoglie oggi circa 100 docenti della nostra università, fondato con un decreto rettorale del magnifico rettore Stefano Bronzini nel giugno del 2020, a 3 anni circa dalla sua origine, ha dimostrato di avere una forte vitalità e una forte capacità progettuale. È infatti noto che abbiamo ricevuto noi del CITEL un progetto ministeriale, l'approvazione ministeriale di 4 milioni di euro, di cui un'IBA è appunto capofila, il centro hub, quindi questo ci fa molto onore. Ora, fra gli altri progetti c'è questo progetto. Il CITEL è infatti un centro con professionalità molto eterogenee e per questo importanti, quindi con un'interdisciplinarità. Abbiamo non solo medici per l'appunto, ma psicologi, biotecnologi, biologi, statistici, informatici, chiaramente, scusa se non ti ho nominato subito, informatici, e poi appunto tutti legali, appunto giuristi, quindi tutti insieme a far sì che progetti di carattere medico-sanitario siano competitivi in un contesto internazionale. Questo progetto della nostra università è un seed, infatti è un seme che oggi sarà descritto e che ha generato un progetto ancora più grosso, ancora più focalizzato sulla diagnosi precoce, sulla terapia precoce e quindi sul miglioramento della prognosi dei pazienti settici. Dicevo, solo in un centro interdipartimentale con numerose professionalità che guardi alla nuova sanità si può riuscire oggi a fare progetti, ripeto, competitivi in un contesto internazionale. E io ringrazio molto il professor Michael Lustler, Giuseppe Calamita, il professor Caivano per aver dato luogo appunto a questo incontro, che è un incontro informativo e dà appunto l'idea dello spessore della ricerca che oggi viene fatta nella nostra università e appunto nel CITER. Ringrazio molto e passo la parola al professor Caivano.
Buongiorno a tutti, io in primis vi porto i saluti del magnifico rettore dell'Università di Bari, e do il benvenuto a tutti quanti, benvenuti all'Università di Bari. Progetto H-Seeds a livello d'ateneo è un'iniziativa che ha puntato principalmente a due obiettivi. Il primo obiettivo è stato quello di Il primo obiettivo è stato quello il primo obiettivo è stato quello di aumentare la competitività dell'Università di Bari nella dimensione europea, quindi spingere i ricercatori dell'Università di Bari a proporsi in risposta delle colle europee. Il secondo obiettivo è stato quello, il tentativo di sfruttare quello che è uno dei vantaggi più grossi di un ateneo come l'Università di Bari. L'Università di Bari è un ateneo generalista, all'interno 19 dipartimenti di ricerca, 42.000 studenti, 3.000 tra personale tecnico-amministrativo e docenti, ma soprattutto copre ad oggi tutte le aree del sapere. E questo per noi è un grande vantaggio che va sfruttato. E va sfruttato perché oggi, guardate, Horizon Europe, no? Che è il più grosso framework di ricerca mai varato dalla Comunità Europea, ci porta a confrontarci con quelle che lui stesso definisce le grandi sfide. Quindi, piuttosto che incentivare singole discipline, la cosa che ci chiede è capiamo come queste discipline possono contribuire a vincere queste grandi sfide. E le grandi sfide sono qualcosa di complesso e la complessità non la si può affrontare nella dimensione della singola disciplina, ma richiede trasversalità. Allora, un po' per sfruttare questo grande vantaggio, con l'iniziativa Progetto H-Seeds abbiamo inteso, come dire, spingere verso la collaborazione tra docenti che ognuno eccellenza nel suo settore, magari non riuscivano a capire l'uno cosa fa l'altro o si guardavano in cagnesco, quindi è stata la prima iniziativa questa che ha portato a parlarci, realizzando così realmente, Giuseppe lo diceva prima, una contaminazione di saperi, di metodiche, di linguaggi, di culture, e ci siamo resi conto che io, Giuseppe e Angelo, che prima ignoravamo cosa l'uno facesse rispetto all'altro, in realtà siamo sinergici. Con questa iniziativa abbiamo realizzato una collaborazione trasversale tra 1200 docenti rispetto ai 1500 dell'Università di Bari, quindi un risultato enorme, se considerate che in ciascuno delle proposte SITS finanziate sono presenti almeno 3 docenti per ciascuna delle 3 aree che al minimo devono essere previste. Quindi abbiamo, come dire, imparato a dialogare ad oggi e ci siamo presentati. Ora lo step successivo che dobbiamo fare è far sì che questo dialogo si traduca in risultato. Parlavamo di sfide europee. Le sfide europee, dicevo, richiedono complessità, ma soprattutto richiedono progettualità capaci di generare impatto. Oggi il concetto di impatto è fondamentale, sociale, economico. E l'impatto, tradotto in soldoni, cos'è? L'impatto è ciò che della ricerca mia, di Angelo, di Giuseppe, i cittadini ricorderanno tra 10 anni, perché significa che avremo dato loro un qualcosa che ha migliorato la loro qualità della vita. E quindi è questa la direzione in cui andare. In particolare, lasciatemi dire anche che questo progetto rappresenta proprio la quintessenza di tutto ciò, della collaborazione, dei risultati, delle grandi sfide e, non in ultimo, dell'innalzamento della qualità della vita dei cittadini. Io chiudo qua, benvenuti nell'Università di Bari, grazie a tutti e buon lavoro.
Possiamo passare alla parte delle presentazioni scientifiche in attesa del dottor Montanaro. Colgo l'occasione per far presente e ringraziare tutti coloro che sono collegati in remote, perché l'evento è trasmesso in diretta streaming, quindi un saluto a tutti coloro che ci vedono da casa. Permetteteci un, direi, affettuoso ringraziamento e veramente congratulazioni alla dottoressa Lofino con Ambitel News, che veramente in maniera molto accurata e professionale sta gestendo da diversi giorni tutta la parte di preparazione dei file, della trasmissione, i tecnici bravissimi, quindi per noi è anche un'occasione veramente bella, perché ci dà la possibilità di divulgare, di disseminare. Questo è uno degli intenti, come diceva il collega professor Caivano, principali di questi progetti, quindi meglio di così, veramente siamo felicissimi per questo, quindi ovviamente ringraziamo tutti e quindi io direi possiamo già passare alla prima delle 3 sessioni dell'evento, sarà moderata dal professor Vacca.
Okay, good morning, and I asked Dr. De Santis to be here and talk about Sepsi, clinical translational study uncovering a new potential therapeutic target. Please, Dr. De Santis.
Non si preoccupi. Good morning, and thanks to Professor Vacca to introduce me, and thanks also Professor Calamita for inviting me to speak about our topic in this project of Horizon Europe Seeds. According to the National Institute for Health and Care Excellence guidelines, sepsis is caused when the body immune system becomes overactive in response to an infection, causing inflammation which can affect how well other tissues and organs work. Sepsis is a syndrome formed by different body dysfunctions, and these dysfunctions evolve in a predictable way. Sepsis also has a high mortality rate and a variable clinical presentation. Today in the world there are 49 million of cases of sepsis each year, and of which 11 million of these deaths. When sepsis is recognized early, patients can be quickly given the right treatment. However, since signs and symptoms can vary and may be susceptible, sepsis may be diagnosed less often than it is actually present. If we consider the sepsis spectrum, we can consider the first the systemic inflammatory response syndrome that involves in sepsis, SARS plus infection, and then in severe sepsis and in septic shock. Different are the risk factors for sepsis: the suppression of immune system, the age of the patients, very old or very young, the patients that received an organ transplant or surgical procedures, the indwelling devices, and the presence of chronic disease such as renal and liver failures, diabetes, or autoimmune disease. The role of our unit, that is the Med unit coordinated by Professor Vacca, in this project is to identify the coagulative aspects and the immunothrombotic processes associated with endotheliopathy, with the aim of correlating these parameters with the clinical evaluations of septic patients, to define the type of infection in septic patients, and to perform an early identification and stratification of prognostically unfavorable factors useful to create an efficient management of the enrolled patients. At first, we obtained the approval by the Ethic Committee, and all enrolled patients provided informed consent according to the Declaration of Helsinki for this pilot observational study. And we create a database containing different informations about enrolled patients. Demographic informations, the information about the origin of the septic infection, and the outcome of the patients enrolled, such as the intensive care unit transferred. This database contains also the information about the laboratory parameters at basal time and at a different time points, such as neutrophils, lymphocytes, monocytes, and above all lactates and procalcitonins that are the two important parameters that correlate with the progression of septic process. In this database we have also the clinical parameters and the comorbidities of the enrolled patients, such as diabetes, hypertension, and the presence of COVID. Then we perform a stratification of the enrolled patients according to different clinical variables and tools that are commonly used for a sepsis screening, such as the Quick Sequential Organ Failure Score, or QSOFA, that are formed by three criteria: the hypotension, systolic peripheral blood, the altered mental status, and tachypnea. And when two or more of these criteria are present, this suggests a great risk of a poor outcome for septic patients. We have also the other criteria, such as sequential organ failure assessment, or SOFA, National Early Warning Score MUSE, and Modified Early Warning Score MUSE. For each patient enrolled, we collect serum and we create a library of available serums for these patients. And finally, we identify potential biomarkers in patients hospitalized for sepsis or septic shock, useful for new pharmacological targets. To obtain a more efficient management of patients with severe infection, we enrolled the patients and we stratified the enrolled patients in relation to the type of the microorganism that induce septic condition, such as Streptococcus, Staphylococcus, Enterococcus, Klebsiella, Candida, and Pseudomonas, the type of the antibiotic therapy that patients received, the other drugs, or the predisposing factors, such as the use of antibiotics in the previous 3 months. Concluding, the results obtained so far allow us to collect and categorize new elements useful for early warning and timely diagnosis, to introduce the personalized medicine approach, validating diagnostic parameters, SOFA, SOFA, Muse and Muse, in relation to the clinical phenotype of enrolled septic patients, and to identify more effective diagnostic and therapeutic strategies. An important question is the limits of this study, that is a pilot study, H-Seeds, the narrow sample of around patients, and the potential number of patients whose sepsis condition has been underdiagnosed to date. So we thank the operative unity of internal medicine Baccelli with Professor Vacca, Dr. Antonio Selimando, all my laboratory staff, and special thanks to Professor Giuseppe Calamita.
Thanks, Dr. Lusanti. We have time for 2 questions, short questions about clinical managing of sepsis. Which are systems that you will use to correlate new parameters with clinical parameters you showed in the slides? Do you perform some statistical analysis?
Yes. At the beginning, we performed to date only a descriptive analysis to observe the obtained data, and we found that these data are very important to stratify septic patients in the less time possible to become a new therapeutic approach, a new therapeutic discover or drugs to reduce the time in which the patients are treated.
Okay. Yes, we can move to Giuseppe Calamita. It's my pleasure to introduce Giuseppe Calamita, the head of this project. So, Giuseppe will speak about the white blood cell, aquaporin 3 and aquaporin 9, new prognostic biomarkers and drug targeted sepsis. Please, Giuseppe.
Thank you very much. Actually, I have met the chief is Angelo, because Professor Vacca, because he had the original idea to create this nice and very original, I would say, timely center, which is this interdepartmental center of telemedicine. So while the file is prepared, I would like just to tell you that the sense of this talk regards aquaporins, because this family of membrane channels are everywhere in the body, as we will see, and in the last years we are finding that they are also involved in the inflammatory response, because sepsis is known to induce a strong inflammatory response. Actually, it is a dysfunctional inflammatory response. So there is a double interest regarding the pharmacological meaning, relevance of these channels, because thanks to people like Dr. Michael Rutzler, who is the CEO of a pharmaceutical company, there are some compounds that blocks in a very potent and selective way these aquaporins. So there are very good premises to develop drugs to fight sepsis. And in addition to that, the other meaning is the prognostic meaning, because we are finding that they are involved in the disease. So, just to tell you very rapidly, most of you know this very interesting family of membrane channels. In the humans there are 13 isoforms, as you can see, based on the functional properties of transport. They are not only water channels, which was the original finding, but they transport many other molecules. Among these molecules, hydrogen peroxide, that, as you know, it is a very important molecule. It is not only a ROS molecule, but it is also a very important signaling molecule that in sepsis, in the inflammation, has an important role. Aquaporins are everywhere in the body, in all of the body districts, where they play important physiological functions, but they are also found to be dysfunctional in a series of diseases. What we will see today regards the expression of aquaporins in immune cells. As you can see, there are several aquaporins found in the cells of the immune system, playing very important functions. First of all, they seem, and there are strong evidences also from our laboratory regarding the function in the cell migration, especially in neutrophils, monocytes, and macrophages, which are really important actors in sepsis, but they are also involved in the phagocytosis, in the activation of the dendritic cells, in the activation of inflammasome, in the longevity of the T cells, and also in allergies. So they have very important roles, and we are finding that they are also important in sepsis. So, of course, I will be short because we can talk a lot, but as you can see, there are several groups in the world working about the role of aquaporin in sepsis, and they are found in several districts, and in each one they seem to have a role during the septic condition. We are concentrating in the last years, as I was telling you, about some immune cells, and just to be a little bit more defined, dendritic cells express aquaporin 9, and we will see what we found in the last years. Macrophages express aquaporin 3, T cells aquaporin 9, monocytes aquaporin 3, and neutrophils aquaporin 9. And the expression of these aquaporins seems to be important also in terms of amount of the expression. And these are just some of the most important works done by other groups in the world. As you can see, aquaporin 3 has been found to be important in the chemokine-dependent T cell migration, thanks to the mediation, to the facilitation of the hydrogen peroxide uptake. Aquaporin 9 has been found to be also important in the phagocytosis, which is something that in sepsis, when of course there is the origin, microbial origin, it is something important. And also in the activation of inflammasomes, in particular aquaporin 3 has been, there are some preliminary evidences, but we are also working on that, regarding the role of aquaporin 3 in the activation of this important inflammasome, which is central in sepsis. So the story started more or less in 2003. We were just talking with a former student, Marcello Chieppa, who was working on some dendritic cells and who was looking at the effects of quercetin. As you know, it is a compound having strong anti-inflammatory effects. And I just told Marcello, please have a look to see if any aquaporin is somehow modulated in this anti-inflammatory action. So he did a series of preparations and went to see the cDNA microarray analysis results. And we found that very interesting results. First of all, we found that aquaporin 9, the expression of aquaporin 9 after stimulating the cells with LPS. LPS is, as all of you know about this compound, which is able to trigger a strong inflammatory response. And one of the first events consists in the activation of the dendritic cells. And we found that after 6 hours of treatment of LPS, a huge expression of aquaporin 9 in dendritic cells, which accompanied the maturation. And it was something in the early phase, because after 24 hours, the expression was going down. And I will not take much time, but of course we also went to see that in some lymph nodes, and we found some, as you can see, some interesting results. So since we have aquaporin 9 knockout mice, which is a good model to check the importance of aquaporin 9 in any kind of condition we might decide to observe, and we went to see what happens to mice after the induction of LPS. So we did an in vivo study. We did this with some colleagues in France, in Nantes, with Angela Tesse, and we found something very interesting. I will not take much time, but the message, the main result was that after treating the animals with LPS, we found an increase of the expression, actually, yes, of course, an increase of the expression of pro-inflammatory interleukins. And when aquaporin 9 was not there, so in the aquaporin knockout mice, the release, the production of these interleukins was strongly reduced. So somehow aquaporin 9 is important in making, inducing the expression of these pro-inflammatory interleukins. So we went further. So we went to see the role of aquaporin 9 in the maturation of the bone marrow dendritic cells. And again, we found that also the release of some chemokines somehow was related to expression of aquaporin 9, because when it was not there, the protein, things were not occurring. Sorry. Okay. So we went also to do additional studies in terms of real-time PCR, and we went to see a series of compounds. Just to tell you, to be short, we found, as I was telling you in particular, that the expression of the pro-inflammatory interleukin 12 was reduced. And on the contrary, we found that the expression of interleukin 10, which is known to be an anti-inflammatory interleukin, was increased. So the pro goes down if aquaporin 9 is there, the anti-inflammatory interleukins goes up. So this is something also quite consistent. And we also found the expression, we went to see the expression of receptors mediating the signals that control the migration of the immunocells in the secondary lymphoid organs. And we found that when aquaporin 9 was not there, this migration was reduced. So seems to be important for the pro-inflammatory interleukin production and to the migration to the secondary sites, so in the lymph nodes. So this is essentially the first conclusion. We kept working on that, and thanks to Angela Tesse, she's at the University of Nantes, and thanks also to Michael Rutzler, we did a nice work, again taking advantage about the aquaporin 9 knockout animals, and we went to induce in these animals a septic shock. Actually, we induced the endotoxemic shock, to be precise, and we used a lethal dose of the work was done in France, and it was a nice collaboration. So we used the lethal dose of LPS, and we went to see what happens. We went to study the survival curve in the wild-type animals versus aquaporin 9 knockout animals, and we found that with this lethal dose of LPS, 20% of the animals survived. So this is something really important as observation. And mice that were dying, they had a delay. So it seems to be important in the endotoxemic shock, aquaporin 9. Again, to be short, because we can talk a lot, but for this event, I think what it is important is just the synthetic message. We tried to understand at what level aquaporin 9 was somehow involved, and we went to measure by electronic paramagnetic resonance the production of nitric oxide, because it is known that nitric oxide is produced in the early stage of the LPS-induced endotoxemic shock, and we went to see that in liver, kidney, aorta, heart, and lung. Because it is known that LPS induces systemic dysfunction, so practically all of the organs are touched. And we found that the production of nitric oxide was not the same. It was reduced in the aquaporin 9 knockout mice after treating the aquaporin 9 knockout mice with LPS versus, of course, the wild-type mice. And we also went to see another. Of course, we used several kinds of approaches. I will not go through all of these images, but they are very clear, because we also went to see the inducible nitric oxide synthase, and by using immunofluorescence, and we found that this enzyme was also reduced. So the expression of this enzyme was reduced. We also went to see the cytochrome oxidase 2, and we found the same thing. And also this enzyme has a role in the early stage of the endotoxemic shock. We went also, because it is known that the LPS binds to TLR4 receptor by inducing a series of steps, the migration of the NF-κB P65 unit in the nucleus, and this is known to induce the expression of a series of pro-inflammatory genes. So just to be short, we went to measure the amount of P65 reaching the nucleus, and we found that when aquaporin 9 was not there, the amount of P65 reaching the nucleus was much lower. That could explain why a series of pro-inflammatory genes were not expressed and they were reduced. And that gave us some information about the molecular mechanism. And we also went, of course, to look at the superoxide anion, and we found the same thing. Also, O2 minus, it is known to be produced in the first part of the endotoxemic shock. And we also went to see this by electronic paramagnetic resonance. We went further because, thanks to Michael Rutzler years ago, synthesized, by starting from a library of synthetic compounds, found HTS13286 being a very strong and selective inhibitor of aquaporin 9. So we took advantage to do the same study in vitro by using the FAO cells, which are hepatoma and rat hepatoma cell line, and we found essentially the same results we got in vivo by using the aquaporin knockout animals. So by blocking aquaporin 9, you get the same results. And this is important because HTS could be a compound that could help to develop drugs, and these are essentially the second type of conclusions. As you can see, I will go very fast. So we could understand that it is important the early stage of the response that involves the NF-κB pathway. Of course, further work is needed to have additional information. I will not go through, but as you know, all of the researchers are a bit crazy in the sense that they have to stimulate the brain to make some hypotheses and to check if they are correct or not. So this is more or less what we are thinking about the way aquaporin 9 is involved. in what we saw. I cannot go in detail, but you know, this is a working hypothesis, an hypothesis for the moment. So we went further, and we did recently a nice work that was published last year. This is something also quite, I would say, transversal. I don't know if we can use this word in English, but anyway, we put together a series of competencies from London, Bari, Nantes, and of course Lund, Sweden. Again, thanks to Mikael, who is, you know, doing more and more about these compounds. A new compound inhibiting aquaporin 9, RG100-204, was checked on a model of cecal ligation and puncture, which is a model, an animal model, a murine model of polymicrobial sepsis. And so we started by checking this compound on human recombinant aquaporin 9, and we found, this is work we did also with some colleagues in Germany and another colleague in Sweden, Urban Johansson. We found that we could confirm that it was working quite on the recombinant protein, and we could see that it was inhibiting the hydrogen peroxide transport, the water transport, and the glycerol transport of aquaporin 9. And this is something showing how the model is made. And again, just to be short, we found some very important, we made a series of important observations. Here we are showing just part of those observations, because the paper has about maybe 20 figures, something like that. First of all, we found that if we block aquaporin 9 by using RG100-204, the NLRP3 inflammasome is not activated, and that might explain why when the animals are treated, pre-treated and post-treated, or post-treated with the inhibitor, the dysfunctional, some organs like heart and kidney, which are strongly affected in this model, there is a strong amelioration. And as you can see here, we can say that we found, I did not go through all of the data, but anyway, there is an attenuation of a series of symptoms, hypothermia, systolic and diastolic cardiac dysfunction, and also an attenuation of the renal dysfunction. We also found that markers typical of this condition improved a lot. And the interesting thing was that we could see the effect even by administering compound 3 hours after the CLP, and also the fact that the inflammasome NLRP3 was also involved. Just to finish, these are all of the people involved at our department, and soon we will have among people contributing to this work the colleague Rosangela Cardone, the PhD, I would say the doctor, because she's already over as PhD, Daria Di Molfetta. Thank you to Professor Svelto, who is the seed of our work, and people, as you can see, at the other departments at UNIBA, and people over, I would say, the Europe. A big thank to Mikael Rutzler, because we could do many things thanks to Mikael. There is nice collaboration, and I thank you for your attention.
Thank you, Giuseppe, for your brilliant speech. We have time for 2 questions. Please.
Thank you, Giuseppe, for your nice results. I have a very simple question. What would be the advantage for cells to increase the expression of aquaporin 9 after exposure to inflammatory agents or physiologically? Why this would be advantage?
It's very simple. I mean, based on what I showed that based on our hypothesis, for sure it is known that after you treat either cells or animals with LPS, which is known, not the only one, but several other molecules are present, there is a production of hydrogen peroxide, which is actually the hydrogen peroxide comes from the superoxide anion, which is produced after the LPS stimulation. The superoxide anion gets out of the cell, and it is known. I mean, actually, it is produced out of the cell, and after the superoxidase dismutase transforms O2 minus to hydrogen peroxide. So you need that hydrogen peroxide gets into the cell to trigger to help, to reinforce, I would say, the pathway that brings P65 to get into the nucleus. To be short, in that kind of reaction, you need hydrogen peroxide getting into the cell. And aquaporin 9 is a very good peroxyporin. So the reason why, but you know, you don't need to increase the expression because aquaporin 9 is already there. You need a facilitated pathway to have hydrogen peroxide getting inside. So this is our hypothesis. But of course, since this channel conducts also water and glycerin and some other molecules, we are not 100% sure that it is important for conducting other molecules. For the moment, based on some results we had and also based on other groups, it seems that it is important because of as peroxyporin. But you know, maybe something else is also there. Thank you.
Okay. Giuseppe, do you have proofs that blocking aquaporin 3 or aquaporin 9 is more potent than blocking central cytokines such as TNF-alpha or IL-6?
You know, for the moment, what it seems to be clear, you know, it's coming up, is that these aquaporins, both aquaporin 3 and aquaporin 9, somehow are involved in the process that brings to the production of the interleukins. So, to answer your question, maybe you know better than me because you are also a clinician, but they are two different levels, and for some reasons we are more interested about aquaporin 9 because in humans aquaporin 9 has two predominant expressions, liver and neutrophils. So it seems to be a good candidate to develop a drug because you can touch only some cells, and aquaporin 3 actually has a larger expression. But I think there are two levels, and of course, you know, once you know that one level, the one through the aquaporins, works fine, maybe it could be easier to see by using models if it is more convenient, more effective, one or the other one. But for the moment, I think it's important to see to explore if this pathway could be, this strategy could be successful.
Monica, please.
Yes, thank you for your presentation.
Thank you to you, Monica.
Just a brief question and out of curiosity. You showed that the survival rate was approximately higher, 25% in your
25% of the survived.
Okay,
survived. The other ones, they survived means that they were able completely, okay, which was really shocking for us because normally, actually, by thinking about what it was done,
right.
I'm joking, but I told Angela because she said, let's use 40 micrograms per kilogram of body weight. We could use 10 micrograms having the same kind of effect. So what I'm trying to say is that the dose was important, and we were really surprised that they were able to survive, to recover even from the symptoms.
Following to this, just out of curiosity again, did you measure the blood pressure? Because usually it's high, the consequences.
You know one thing, you know,
yes,
Assunta,
is
Professoressa Potenza is there, and that was our idea, and that's why we also, you know, we tried to do something. There is a problem, and this is something maybe generally in Italy. The problem is that when you do these experiments in Italy, you cannot go over a certain level of pain. So you have to stop the experiments before you get to the end. Okay. So this means that it is very hard to do this. That's why in France things are a little bit less severe. But you know, it's not logistically easy to do the measurements important that you are suggesting, because you know that was the problem. So we cannot do much. That's why also Michael Rutzer with Apoglitz committed the work to some private companies out of Europe. In Croatia, for example, right now we are repeating the experiments in Brazil because things are much easier. So that is the problem, the obstacle. Thank you.
Okay. So, a question?
Just a curiosity.
With big pleasure.
In this model, do you know if the skeleton is involved? Yes.
You know, we normally in this model the skeleton is involved for sure. There is some work done. We plan to do things with the bone. The problem is that again we have here colleagues being in the ethical committee. The problem is that here in Italy it is almost impossible to have the full model, because you have to stop your experimentation soon, because when you induce the endotoxemic shock, you have a strong reaction. So, but I know some people are doing that, but this is among the things that we are going to see. Actually, of course, you know, from the bone you can take the bone marrow, which is something else. But in any case, also because aquaporin 9 is expressed in the bone, in the chondrocytes. Yes. Okay. Thank you.
We have to move to Dr. Colomeo. On long non-coding RNA and sepsis predictive diagnostic and prognostic methods. Please go.
Good morning and thanks to be here and to give me the opportunity to talk about these really interesting and powerful, in my opinion, molecules that are the long non-coding RNAs. Long non-coding RNAs are non-coding transcripts longer than 200 nucleotides, with a very specific expression that is cell and stage dependent. They have a broad range of activities in cells, both in physiological and pathological conditions. This is because they can interact both with RNA and DNA molecules, but also with proteins, thanks to their three-dimensional structures. The main function of the long non-coding RNAs is regulating the gene expression by acting both at transcriptional level, for example, guiding specific proteins such as chromatin modifiers to specific genes, and at post-transcriptional level, for example, acting as competing endogenous RNAs that can sponge microRNAs. Due to all these functions, an alteration in the gene expression of the long non-coding RNAs is involved in many diseases, including sepsis. Indeed, in the last 10 years, increasing literature is analyzing the role of the long non-coding RNAs in sepsis pathogenesis. However, there are many points still to be clarified, and this is because the scene is really complex, especially because there are some molecules that can have a context-dependent role. This is, for example, the case of the long non-coding RNA TAG1 that can display both an anti-inflammatory and pro-inflammatory role depending on the tissues in which it is expressed and analyzed. In sepsis patients, hyperinflammatory state coexists with immunosuppression, as we know, and long non-coding RNAs play a pivotal role in both the states. For example, the long non-coding RNA NEAT1 can activate the toll-like receptor 4 signaling pathway and hence having stimulating the inflammatory state. On the other hand, the same molecule can also have an opposite effect and, in detail, can increase the apoptosis of T cells, hence displaying an immunosuppressive role. Conversely, there are also long non-coding RNAs that can increase the proliferation of immune cells, such as the monocyte-stimulating MALAT1. One of the reasons of the high mortality of sepsis is also the sepsis-induced organ dysfunction, and also in this case long non-coding RNAs play really important roles. For example, in the sepsis-induced acute lung injury, it has been reported the involvement of the upregulated MALAT1 and NEAT1. Due to their roles in sepsis pathogenesis and also in sepsis-related organ dysfunctions, long non-coding RNAs has been investigated for their potential as biomarkers in sepsis. This is because there are differences in their expression levels between sepsis patients and healthy people. Moreover, long non-coding RNAs can be really easily analyzed by starting from blood samples. Indeed, blood includes both plasma and serum, giving the opportunity to analyze circulating long non-coding RNAs and also cells. The analysis can be performed by RT-qPCR techniques that are really easy to be performed and moreover are less time-consuming compared with other techniques, such as, for example, the microbial culture. For sure, microbial culture is really important and essential for sepsis diagnosis and treatment. However, it can require a few days to give results and sometimes can also give false negatives, especially when antibiotic treatment has been already started. Among the long non-coding RNAs investigated for their role as potential biomarkers in sepsis, in our Horizon Europe Seeds project, sorry, I have some problem. Sorry.
You can use the other one.
Yeah, this one?
No.
Okay. In our, maybe too high, in our Horizon Europe Seeds project, we are investigating 3 molecules that are MALAT1 and NEAT1, that are the most investigated long non-coding RNAs for their involvement in sepsis pathogenesis, and PBT1. This is a really interesting long non-coding RNA because it is involved in several diseases, including cancer and sepsis, where it shows a pro-inflammatory role. Moreover, MALAT1, NEAT1 and PBT1 act as positive regulators of the node-like receptor protein 3 inflammasome that has, as Professor Calamita told, a central role in the inflammatory state in sepsis patients, and that is a target of our analysis also because it is involved in the aquaporin pathways. There are two independent studies in the literature analyzing the role of MALAT1 as potential biomarkers in sepsis. In these studies, the expression level of MALAT1 was evaluated analyzing plasma samples from sepsis patients and healthy control. As you can see from the image, first of all, the authors revealed an increased expression level of MALAT1 in sepsis patients compared with healthy control. Moreover, analyzing the ROC curve, MALAT1 was revealed as a significant predictive marker for indicating the sepsis risk and for discriminating sepsis patients from healthy people. MALAT1 had also good values in predicting the 28-day mortality in sepsis patients. Similar results were also obtained for NEAT1 and PBT1 in two different studies. Both these long non-coding RNAs were upregulated in sepsis patients' plasma compared with samples from healthy control, and NEAT1 showed also good value in predicting the sepsis risk. Moreover, NEAT1 and PBT1 were associated with a worse prognosis of sepsis patients. All these studies underlined the potential of these long non-coding RNAs as biomarkers in sepsis, underlying their correlation with increased sepsis risk and poor prognosis. Moreover, the expression levels in plasma of these long non-coding RNAs were also associated with some parameters already used for the sepsis diagnosis, such as, for example, the SOFA score. However, also these studies have some limitation and critical points. First of all, the expression of the long non-coding RNAs were evaluated once for each patient, make it impossible to understand if long non-coding RNAs can also use as follow-up markers. As a second point, all these studies used as control healthy people. This is for sure a good way to evaluate the potential of the long non-coding RNAs as diagnostic biomarkers in sepsis. However, in this way, it's not possible to analyze if long non-coding RNAs can also be used to differentiating between patients with sepsis and patients suffering from other immune-related diseases. Hence, further studies are for sure needed to address this point, as well as further studies are needed to better understand the molecular mechanisms behind the increased expression levels of long non-coding RNAs in sepsis. And for sure, it will be really important to increase the number of the analyzed patients. Finally, long non-coding RNAs in the study that I showed you were individually analyzed for their potential as biomarkers. Instead, in our Horizon Europe Seeds project, we are analyzing MALAT1, NIAT1 and PVT1 for their potential as a combined biomarker in sepsis patients. To this aim, we extracted blood samples from sepsis patients and healthy control, starting from 500 microliters of plasma. We extracted the total RNA, quantify and retrotranscribe it. MALAT1, NIAT1 and PVT1 expression level, I am talking about MALAT1, NIAT1 and PVT1 circulating molecules that are in the plasma, were evaluated by RT-qPCR experiments using a pool of RNA from healthy people for healthy control as calibrator. The data we obtained will be combined with data obtained from the analysis of the expression of aquaporins and also with the clinical parameters in order to identify a combined profile of diagnostic and prognostic biomarkers for sepsis patients. I would like to highlight that this is a pilot study for sure, and we need to increase the number of the analyzed patients that are at the moment 50. The data analysis is still in progress. I would like to thank you for your attention and thank all the people that are involved in this project. First of all, our PI, Professor Calenta.
Thanks, Lauren, for this brilliant speech that is open to 2 questions. Please.
What is the origin, which cell type is producing these long non-coding RNAs?
Probably they are originated from immune or inflammatory cells. So they can have different origin. For example, they can be secreted in the exosomes and so released in the plasma, or they can derive from cell debris. So from apoptotic cells, but always cells involved in the immune response.
Did you plan to study the respective mRNA that are blocked by this long
yes, for sure. It's in the future for sure, because the main role of these long non-coding RNAs is sepsis is linked to the role as competing endogenous RNAs. So the microRNAs have a pivotal role as the long non-coding RNAs.
Okay, thanks. Please, Professor Bilancia. I'm happy to introduce Professor Bilancia, biostatistician of our department in the computational landscape of sepsis. Please, Mr.
Thank you. Thank you very much for this very interesting event. And before talking of the computational landscape of sepsis, a few words about the number of sepsis to explain why sepsis is the most serious emergency today. 49 million individuals relating to sepsis and 11 million deaths worldwide. And 1 death over 5 per year is associated with sepsis. We are at the same level of cardiovascular mortality. And 1/6 experience long-term sequelae that worsen quality of life. And what about Italy? Age standardized death rates are increasing remarkably, both for males and females. For example, we had about 80,000 death for sepsis in 2003, rising to about 49,000 deaths in 2015, from 3 to 8% of all deaths. But some more realistic estimates are about 20% of all deaths related to sepsis. And around 17% of new sepsis are community acquired, out of hospital, out of wards, out of ICU. And ICU acquired sepsis occurs frequently in Italian ICU patients, about 22%, and are associated with a high case fatality rate, high length of stay, and a high degree of multi-resistance. Well, a few words on the machine learning workflow. What can machine learning do for sepsis? Two words. Supervised learning, we learn from past example and try to generalize to future patients. Unsupervised learning, we try to characterize the patients, to individuate distinct phenotypic groups with distinct outcomes. And two distinct problems. First big problem is that of early warning. Timely intervention is critical. Early diagnosis of sepsis is critical in reducing mortality. Delays of more than 6 hours increase in the diagnosis, increase significantly mortality, and increases the risk of progression to severe sepsis and septic shock. You can see this is the typical workflow of an early warning system. We have both structured data, clinical data, and but we have also clinical annotation. So we have the interaction between traditional machine learning methods, such as deep learning, and methods involved with natural language processing, which is that part of machine learning which deals with natural language, interpret natural language and makes prediction using natural language. One example, the SIRA algorithm, sepsis early risk alert. Two kinds of data, a supervised system. We have we have both clinical data, but we have also clinical annotation, and the SIRA system allows to predict sepsis in the 88% of cases 6 hours before the onset of sepsis, against the 58% of inexperienced clinic. On the right, you can see a ROC curve. A ROC curve, the classification is better when the ROC curve is near the upper left angle. So you can confront, for example, the goodness of classification by the SIRA algorithm compared with traditional scoring systems such as SOFA, MU, SIRS, and other. The SIRA algorithm does better. Another early warning system, the Target real-time early warning system, a very large multicentric study published on Nature Medicine, about 600,000 patients across 5 hospitals, and in particular a large cohort of patient risk extracted from this very big super cohort. In the alert by the TREB systems, we have that in-hospital mortality is reduced of about 4% if the alert is confirmed in the first 3 hours. We have an adjusted risk difference of about 3 points and adjusted relative risk of minus 18 points. This confirms the usefulness of this early warning system. And another problem is unsupervised, is sepsis characterization. Can we characterize the patient phenotype? Sepsis is a disease characterized by a large heterogeneity. The clinical trajectory is often unpredictable. Can we determine in advance the sepsis severity? For example, we have a well-established result, 4 groups of septic patients. The first group is that of neutropenic sepsis with low white blood cell count. The second group is typically that which has the lowest SOFA score, has the highest survival rate. The third and the fourth group are those groups in which you have the worst performance, in particular the fourth group, in which you have significantly prolonged prothrombin time, which is probably linked to epithelial dysfunction and then immunothrombotic phenomena, which are, for example, investigated by the group of Angelo Vacca and Antonio Solimando. And another important problem, another important diagnostic problem, the identification of new biomarker for the prediction of mortality, which are the most important papers that I can cite, which are, for example, the fact that an important independent predictor of mortality is tumor necrosis factor alpha. This is a well-established result. Blockers of tumor necrosis factor alpha are used since many years and also the Inflamix SIF2, which is a classifier based on 29 messenger RNA selected site, which has high sensitivity and specificity in predicting the severity of sepsis and the onset of septic shock. And, for example, the prediction of sepsis mortality from blood metabolites. This is an active area of research. To conclude, there is a lot that machine learning can do for sepsis, both in the area of early warning as well as in the area of biomarker discovery and diagnosis of sepsis, because there are a lot of unmet needs that still needs to be satisfied. Thank you for your attention.
Thanks, Massimo, for this talk. Please, Alessandro.
Just a curiosity, you highlighted an important point about unstructured data. And,
about?
Unstructured data, the approach to analyze unstructured data, text mining and
yes.
all that data contained in reports, in other documents. Is there a literature, a well-defined approach merging structured data analysis with unstructured one?
Yes, the Zero algorithm do that. The Zero algorithm merges clinical data and text data, clinical annotation. Yes, there are, the Zero algorithm is a prominent approach merging clinical and laboratoristic data and text data, clinical annotation. And merging these two aspects, you have an increased accuracy in predicting the onset of sepsis. You have 88% 4 hours before the onset of sepsis, against 56% from an experienced clinic, because initial symptoms of sepsis are generic, are not specific of sepsis, and sometimes the onset of sepsis is abrupt, is sudden. So you must distinguish sepsis from other more benign conditions.
exactly hidden information in unstructured data? There are a lot of hidden information.
Yes, there is a lot of information in a structure that need to be combined with clinical, laboratoristic, immunogenetic data, which sometimes are often difficult to obtain, requires more time, and sometimes the septic event has already been present in the patients. And sometimes it's too late.
Thanks, Massimo. So I ask Nicolò Mucciaccia to speak about the legal protection of new algorithms in healthcare. Please.
Niente, è partita la presentazione di prima, un attimo solo.
Sì, sì, quello.
Anche lei imposta la presentazione così.
Ok, grazie.
Ok.
Grazie.
Giusto un attimo.
Yeah. Okay. Good morning to everyone. Thank you for your kind hospitality. And my presentation is quite a bit uncommon for maybe the scientists, because it's about the legal protection of the algorithm. That is, our project needs to study from a juridical standpoint, needs to study data in order to, does it work? Okay. To elaborate data in order to make a diagnosis, can I say, there's something to prevent. Okay, so from a juridical standpoint, there are two different problems that are the protection and the exploitation of the algorithm made for these studies. Okay. And there's also a problem of privacy for data, okay? So, always from a juridical standpoint, there's not a definition of algorithm. So, the algorithm can be protected as with a patent, maybe, but we have to think about it. Then there can be the copyright law, because the algorithm can be intended as an instruction, okay, that we give to the machine. So it can be made a parallel with between the algorithm and the software. But actually, the software is something that makes the algorithm executed, and so the algorithm cannot be considered as a software. So if we come back to the patent law, we have to think about the algorithm as a mathematical method, maybe. But the mathematical method cannot be patented if, as such, okay? The mathematical method can be patented if it has a technical effect that is other than the mathematical method itself. Okay, so the algorithm can be protected not as such, but only if it has a technical effect that is an impact on the hardware on which it works. Okay? So, there's another way to protect the algorithm, that is the trade secret. The trade secret has different problems, presents different problems, because there's always the need for a further technical effect to protect the algorithm as a trade secret, not in itself, I mean. And the conditions are that there must be a secret, of course, so the algorithm the algorithm must be kept secret, then it must have a commercial value as it is, okay, as a secret. And then there must be the adoption for reasonable measures to keep the algorithm secret. Then there's also the risk, if we use the trade secret discipline, I mean, that the algorithm could be disclosed to third parties. Think about the unlawful dependent that, or disloyal employee that communicates the algorithm to third parties. Okay, so it can be a problem. Then there's also the possibility to exploit economically the algorithm. So if we make a study and we elaborate an algorithm, the algorithm can be exploited economically. It means that we can transfer the right on the algorithm in itself, or we can make a licensing agreement, okay, to transfer the possibility to use the algorithm with the exclusivity or without the exclusivity, and then it is possible, the licensing agreements make it possible for spin-off companies to be the, let's say, proprietor of the algorithm. And so it can be also useful for university, I mean, and it's our point. Then there's the second problem, that is the problem that concerns the protection of health data, because for the study on sepsis, of course, we must use personal data. Personal data have a peculiar discipline for privacy, because there's the reuse of personal data. There can be the reuse of personal data. What does it mean? The use of personal data, as you know, must be there must be the consent from the patient for the use of personal data according to the GDPR, okay, the discipline on the privacy, the regulation about privacy in the European Union. But the reuse, and that is the point, in order to conduct a study, you use the data that you have collected from patients in order to make a study that is a different use other than the one for which you have collected the data, that is for medical reasons, okay? So the problem is it possible to use the data that you have collected for medical reasons to conduct a study, okay? Or do you need the consent of the patient? It is possible under certain circumstances, of course. So, if the patient gives the consent, it's okay, of course. Then, it is possible, according to the GDPR, that is our discipline, because there are some exceptions that are allowed. These exceptions are reasons of substantial public interest, reasons of public interest in the field of public health, that is health emergencies resulting from earthquakes or food safety, and then it is possible to use the data, and that is this is the point, purposes of preventive medicine, health or social diagnosis, care or treatment, or management of health or social systems and services, that is for care purposes. Okay, so there's also the possibility, and it's always possible to use anonymized data. Okay, so if you collect data, you can anonymize them and use them for other purposes than the one for which you have collected them. So, what's the problem in the anonymization process? Is that anonymous data can be used if are anonymous, okay, because it must be impossible to reconduct the data to the person that has given the data. So, there are always new technologies coming, and so the problem is, what does anonymization mean? Because something that can be considered anonymous today cannot be considered anonymous maybe tomorrow with an oncoming new technology. So of course we have to balance the need for the use of the data, that is for clinical reason or other is for study reasons and so on, with the effective protection of privacy. So what's the GDPR says that there must be a reasonable likelihood of the use of the means to identify the natural person. It means that today there must be a reasonable likelihood that the data won't be de-anonymized. Okay, so that is sufficient for the GDPR, and so you are responding to the law, okay, complying to the law. Then there's the last problem, that is the big healthcare data. It means a set of data that are collected always by also our watches or other stuffs, and they are put together and used to feed an algorithm so that the algorithm can elaborate the data and predict something. Okay? Here we have also the PSI directive, that is the Public Sector Information Directive. With respect to the GDPR, the general principle is exactly the opposite. That is, the freedom in the use of public sector information with some limitations. So, the GDPR says that it's impossible to use the data, but there are some exceptions. The PSI says the opposite, that is, the freedom to use the public sector information, but with some limitations. And there's also a principle of free use at Article 6, and so there must be a combination between the GDPR and PSI when we are talking about big healthcare data. Thank you for your attention.
Thank you for this legal view of the project. Please, Massimo.
Which panel charge a subject risks not respecting the principle you have exposed?
Panel, I don't know, because the GDPR doesn't, it's a regulation of the EU, and in the EU there's not, how can I say, a single discipline for penal, so it's something in our legal system, but I don't know about the penal law. There's for sure a problem of responsibility, civil responsibility, civil liability, and there are fines, there's the possibility to be blocked, of course, from the use of data, and that's it.
Thank you.
Thank you.
Please, Michael.
So if a patient, after giving consent to data use, discovers that they want to revoke the consent, is there an opportunity for the patient to do that? And if the data is already de-anonymized, can there still be any effect?
Thank you for your question. Yeah, of course, there's always the possibility to revoke the consent, but the consent, I mean, you're asking for a consent that I have expressed specifically for the reuse. Okay, so it's always possible to revoke the consent, of course, before that the data are used. Because if the data are used, it's impossible to revoke the consent, but you can revoke the consent, but not when there are the exceptions and limits that I told you before. So you can revoke the consent, but with these limits.
Good, thank you. We have 20 minutes for coffee.
Grazie mille.
You need a technical time of 1-2 minutes.
Okay, no,
Oh, we can start. I think we can start, and we have this second session. The title is Artificial Intelligence as an Extra Weapon to Fight Sepsis, the EIC Pathfinder Project Antisepsis. And it is a real big pleasure to have Mikkel Rutzler here, CEO of a pharmaceutical company named Apoglix. The main interest is to develop drugs against sepsis. And since Michael has a strong aquaporin background, the main goal is to find selective and potent inhibitors of aquaporins, especially aquaporin 9, which is where the important role in inflammatory response in sepsis, in order to develop drug. So some good results are already there. We already found some good compounds. Some of them are already commercially available, and this is very important also for us. So big pleasure, and you can see the title of the presentation, Antisepsis, or How to Address the Challenge of Drug Development in Sepsis. Please, Mikkel.
Thank you, Giuseppe, for the introduction and also for the invitation to come here. It's a great pleasure to be here in Bari. Also, I'm very glad to be here in the nice city of Bari. And so I am going to talk now about how we want to go from your sepsis project that we are starting as a pilot project, hopefully in the future to the Antisepsis project, and to extend this. I am going to start with a very brief introduction about our company Apoglix and about the data that we have in the drug development. And I would then like to briefly touch on why it is actually very difficult to develop a drug in the area of sepsis, and finally then to present the Antisepsis program. So Apoglix, we are located in the very south of Sweden, close to Copenhagen, in an area of Lund that is called Medicon Village. This is a former site of AstraZeneca. They left about 10 years ago, and now there is a lot of startup companies, so it's actually a very nice place to be. We have good interactions there and good possibilities. This is our company, so we are still a very small startup company, but we have a very experienced board, both from the commercial side and also from the pharmaceutical development. Then we have a small operational team, but we are supported by external consultants that help us in specific tasks and have specific expertise in drug development. So I started basically what has now become Apoglix many years ago when I was working in the laboratory of Søren Nielsen at Aarhus University. At that point, aquaporins were considered a new class of drug targets that are potentially very interesting, but basically also were considered undruggable. I have then adapted a cell-based assay to a format that could be used in a high-throughput screening format. Oh, sorry. Many slides at once. And at that point we found some first aquaporin 9 inhibitors, and then also other inhibitors like aquaporin 3 and aquaporin 7 inhibitors. And from that point actually it was still quite a long road of making a lot of compounds and discarding a lot of compounds and making new compounds to actually take these first hits into something that is now something that is drug-like. So to fast forward, we have started to apply the final drugs. Now adding on to what Professor Calamita already has talked about the LPS model. In inflammation, we tried to reproduce the data that Professor Calamita obtained in the knockout mouse model with LPS. We have tested this with an aquaporin 9 inhibitor, and we saw that the inhibitor caused decreases in pro-inflammatory cytokines and increases in anti-inflammatory cytokines. Since the LPS model is not really a bacterial model of sepsis, we then next tested a bacterial model of sepsis together with Professor Christoph Timmermann. Professor Kalmita already talked about these experiments also, but just one important result that we obtained was we looked at the cardiac output of these mice, probably relating to your question of the blood pressure actually. And we did not measure the blood pressure, but through echocardiography we could actually demonstrate that the inhibitor greatly improved the heart function. Here you can see the non-septic mice. This is the heart function of the septic mice was reduced, and then various parameters of heart function were improved, either when the treatment was started at the same time as the induction of sepsis, but also 1 or 3 hours later, which is an important finding and was actually quite unique. We have recently gone on to also study a model of acute pancreatitis together with Professor Bisheng Shen at Wenzhou University in China. In this model, here you can see the pancreas on the top. These are the untreated mice, and these are the mice with the acute pancreatitis, where you can see that the pancreas is disintegrating. And then here you can see lung tissue, where you can see that the alveolar airspace was compacted, and there was also a lot of interleukin-6 expression. The structure of the pancreas was improved by the inhibitor. Also the alveolar airspace and the interleukin-6 was reduced. So what I would like to summarize at this point, we have looked at various animal models that are also different. Experiments were conducted overall, including Professor Kalamita's results now in 4 different labs. So these results definitely seem to be robust and reproducible. Now, however, coming to the difficulties of drug development in sepsis, I would like to share some views of when I go and talk to big pharma and to investors with these results. It's of course their opinions are very important because they are giving the money and they are the ones who are supposed to take this on at some point. So they have opinions like, for example, mice are not humans and sepsis has killed more companies than patients. The first one most definitely being true, and the second one most definitely not being true, but of course both are somewhat or have some truth in them. Talking about briefly about the mouse model, yes, it's true that the immune system and various parameters are very different in mice than in humans. However, on the other hand, one must also acknowledge that basically the animal models are always very suboptimal, and still a lot of drugs have been developed based on these models, even drugs targeting the immune system. Yeah, and the second, of course, it's definitely true that a lot of companies have failed previously, and a lot of clinical trials have failed in the area of sepsis. So why is that? Well, I guess I will not surprise you if I say that the patient population is very heterogeneous. There is differences in the pathogen, site of infection, age, genetics, immune status, you name it. Now, just for one example, TNF-alpha has already been mentioned as an etiologically very important aspect of sepsis. There have been many drug trials targeting TNF-alpha with antibodies. In meningococcal sepsis, septic shock, the TNF-alpha levels are typically very high. In pneumonia patients, this TNF-alpha levels are typically unchanged or rather low. And a study, I think, with almost 600 patients found they measured TNF-alpha daily, and they found that in more than 50% of the patients, TNF-alpha never goes up, and that even includes a lot of patients that died. So clearly, if you think of doing a drug trial in TNF-alpha antibodies, then you would expect that it would be very good if you could only include patients in the trial that have elevated TNF-alpha levels. However, I assume at least that also those who planned those studies were not idiots. It's not easy to do that because of the still actually in place regulatory guidelines for conducting a clinical trial. For the inclusion criteria, you are not supposed to enroll patients based on biomarkers, but you're supposed to enroll the patients based on clinical scores like Apache or SOFA that are validated. Why is that so? Not because the regulatory body is funny, but because those scores are still performing better than single biomarkers. At the same time, the only acceptable primary outcome criteria if you do a drug trial in sepsis is 28-day mortality. Now, as we already mentioned, we have a very old population with a lot of comorbidities. A recent US study suggested and looked at the hospital death in sepsis, and they suggested that only 11% of the patients could actually be saved by optimized treatment. So if you have 89% of the patients dying anyway, it's quite difficult to prove an effect of a drug. So just by pure statistical considerations, it has been calculated by the current hospital mortality rate of 25%. To reach an improvement of 20%, we would have to enroll more than or almost 1,200 patients. And this is for an exploratory clinical trial, so you're at phase 2. So that makes for a very, very expensive phase 2 and with a very high probability of failing. And that's, of course, the reason why the companies and the investors are not very happy to put any money in this any longer. So what do we do in this situation? Somehow we need to find a way out, and what we are proposing as a way out is to run a clinical trial to enroll about 500 patients, which would be done here in Bari and also at the University Hospital in Strasbourg. We would detect or determine a lot of clinical parameters, immunological parameters, along with 19 predefined clinical outcomes. And we would feed this into an AI algorithm. And we would devise an algorithm that can actually predict then the course of each individual patient a long time. This is, of course, I mean, AI algorithms have already reached a stage where they can retrospectively predict the clinical outcome, but so far, at least as far as I know, it has never been used prospectively, and only for one outcome and not for 19. So this is for sure going to be quite challenging, but we think it's actually the right time to try this. And another problem is that, of course, if you want to do this and actually improve therapy, you have to do this in an ethical way, and you cannot leave everything to the machine learning algorithm. You have to use or leave the clinicians in the loop, otherwise it would be unethical to do it. And the final problem is actually we cannot go on and rely on a guideline from the EMA, from the European Medicines Agency, that is from 2006. We have to lobby, we have to involve stakeholders in order to change the guidelines so that we can actually use this new method that we are proposing to develop. So finally, my personal vision for a clinical trial in the future and for drug trials in sepsis would be to have inclusion criteria that are based on new scores that we are going to develop, that are going to perform better, and that allow us to enroll a less heterogeneous patient population. And the second one, that is maybe even crazy when you think where the guideline is right now, but potentially if you think that we can accurately predict an individual's patient's course, then we could also say, okay, this is where this patient should have gone to, now we treated it, now it goes somewhere else. If we were allowed to do that, if we could prove that this works, then we could run much smaller clinical trials with just a few patients essentially, and prove a statistical effect, and then this would make drug trials in sepsis, of course, not only a lot cheaper, but also a lot faster, and we could get medicine to the patient that they need. So, finally, I would like to thank again the organizers, all my collaborators that have contributed data, and also all the people who helped in conceiving this antisepsis program, not the least Antonio Solemando and Massimo Bilancia, I would like to point out. Thank you very much for your attention, and happy to take your questions.
Thank you, Michael. Any question from the audience? Professor Montagnani.
Thank you very much. Very, very interesting, fascinating, I would say. I have just a curiosity about the molecules you develop, and you show, and Professor Carnita showed some results. Just from a pharmacological point of view, how do they work on the target? Is it a reversible, irreversible, covalent? How long does this effect last? Just to have an idea.
Yeah. So I guess Yeah. So the drugs are completely reversible, so it's fast on and off. The pharmacokinetics, it depends a bit actually on the state of the patients, or currently the patients are mice, but yeah, in the septic mice, so the drug is mostly metabolized by the liver. So in these septic mice, it has a much longer, it stays much longer in the blood than actually in healthy mice. So this is actually also a bit of a challenge, since the patients there will also have very different pharmacokinetics, I guess.
Okay, thank you.
Yeah, really inspiring, thanks. I will try also to accomplish some criticism that the actual clinical approach to septic patient has. The problem that I would like to discuss with you is, as far as the literature is telling us, the implementation of the actual guidelines is not really good. So even an advanced state like Germany or France, they do not apply guidelines. Do you think that our approach, our hypothetical approach, may implement, I would say, from 5 to 10% in a general hospital, those skills to apply or try to apply the guidelines?
Yeah, we have of course thought on how to make this at least more likely, right? And I think one critical point is to involve all the stakeholders, like the Global Sepsis Alliance, and then also to involve really methods that you can easily establish in a smaller hospital will be, or are already established even in a smaller hospital. So those are the things that we can do. I think beyond that, well, maybe there is other ideas, but I don't know.
Thanks.
Yeah.
I have a comment more than a question. It's very important to have a target for drugs, but I would like to underline that one of the purposes of this project is also to identify some mRNA or long non-coding RNA that could be connected to aquaporin. So in the future, one possibility could be to detect and to analyze the expression of these long non-coding RNA in order to correlate with aquaporin. And so if we have a drug against aquaporin, then we can cross the information. Because nowadays we know that RNA is a frontier from drugs, but apart from drugs against mRNA or long non-coding RNA, it could be also important to connect the long non-coding RNA with some other molecules that can be druggable. So I think it's very important to, this is our goal, is just to put the molecules into connection, because even though we don't target the mRNA directly, but if we can cross the information, then it would be very important for patient classification and also for therapy.
Yeah, I think it's, of course, actually when we started out with the whole idea, we basically came from the idea that we should connect the specific molecule of aquaporin 9, the expression of aquaporin 9, with the molecule acting on that. And there is examples in, actually right now under clinical development, especially, for example, adrenomedulin is targeted as a drug and as a biomarker. So I think this is actually a winning concept, but it's not really necessary that we connect this, one molecule as a biomarker with also the drug. I think it's very possible we just need to connect the drug somehow with any number of a bundle of biomarkers that have a good correlation with the response. But that is, of course, the winning concept. Yeah.
Thank you. Any other short question from the audience? I don't see any arm. So again, thank you very much, Michael.
Thank you.
Thank you. So we can move, Nicola, scusa, can move to Professor Antonio Solimando, who is with Professor Vacca. We say in Italy the anima, l'anima, so the soul of the clinical part of the sepsis project, and of course of the antisepsis project that came up from the project, the H-Seeds project. So we rely a lot because the clinical competence comes from your group, Professor Vacca, Professor Solimando, Professor Ria. As you can see, the title is Integrated Approaches to Managing Septic Shock, Combining Fluid, Antibiotic, Immune and Genetic Therapies for Novel Therapeutic Horizons. Antonio?
Thanks a lot for the kind introduction, and it's also really nice to share with you some preliminary data. I have to make a short disclosure due to lack of time. I will just share some preliminary data, but taking advantage of the really inspiring lecture, I would say, from the other speakers, I would sketch some roadmaps that inspired us. And in this regard, I would like to point out that so far we have really a plethora of settings of guidelines. We are right now at the 5th edition of those guidelines. The last one is published in 2021. Nevertheless, as I was highlighting, the implementation in the clinics is really poor and scanty. Why is that? My personal agenda today will be limited to 3 points that we are trying to implement in this Pathfinder project. And so basically screening and initial resuscitation, hemodynamics and infection. With this regard, our preliminary data are quite remarkable in my opinion in terms of trying to redefine the sepsis, or try to provide tools that are able to sketch in advance what is really dangerous situation. The hemodynamics is a really unmet need, and I also think that some preliminary data in our hands could help to sketch the infectious agent and better define the cause. In this regard, I would like to start from the fact that in the previous guidelines, for instance from 2016, we had no mention of Q-SOFA, as the speakers before me already told. So the definition was really poor, was scanty. What they did in the current guidelines, as you all know, Dr. De Santis already pointed out, the Q-SOFA is great. I have to admit it helps a lot. Nevertheless, is not enough to define sepsis, is not enough to screen sepsis, as this meta-analysis is telling us. For instance, the SIRS score is much better than Q-SOFA score in screening sepsis. Nevertheless, the Q-SOFA score is much better than the SIRS score to prognosticate patient. So it's a bit confusing. Our preliminary data, and I thank Massimo to help me out with doing that, confirmed that SIRS, NEWS and MUSE in our pivotal cohort of more or less 55 patients and in retrospective validation cohort are much better to quantify the burden of disease, to screen the sepsis in advance, and therefore to help us out to screen timely sepsis and septic shock. So we can say that SIRS is better than Q-SOFA in predicting sepsis, and we are quite optimistic that, sorry about that, that actually also the guidelines are supporting these, because they just published, that the NEWS and MUSE can be reasonably utilized in order to screen sepsis, and they are much better than Q-SOFA. So, a small tiny piece of evidence. Then we moved to another important problem. So we retrospectively reviewed all the ICD coding, because, as Mikkel pointed out, sepsis research projects are really expensive. So we tried to review several charts, several patients' files, and we realized that the ICD coding for diagnosis and definition of sepsis and event screening are totally not sufficient. So what we could do was to review retrospectively just the case in our hospital and in another pivotal hospital, and in 2019, if we use the sepsis 3 criteria to define sepsis, the incidence is 4.1%. Then 2020 came, unfortunately COVID was there, and we could confirm that actually 4.6% of patients had sepsis, and if we consider the septic patient within the COVID-19, 6.9%. We were quite amused that a pivot study in Sweden actually has the same result with a huge, really huge population, a registry. So they estimate 747 patients over 100,000 is the same incidence of cancer. It's amazing for me. I mean, this is just a number, but as a clinician, this is amazing. So if we want to screen, there are no prospective intervention studies in this regard. We have to admit that when we would like to compare the hemodynamic based on our definition, this is really, really, really annoying, because we have really few clinical parameters to screen these. For instance, capillary refill time, that is sketching the hemodynamic compromised patient. And Massimo already told us that the cluster 4 of patient, the one that is developing the most harmful septic shock condition, the one that is imbalanced in terms of immunothrombosis, is dysfunctional. Why is that? In our opinion, due to the endothelial dysfunction. This is also confirmed by several clinical trials. For instance, the Andromeda shock study on 424 patients, they pointed out, despite the level of evidence is low, that for adults with septic shock is suggested to use a really basic clinical parameter, a really cheap clinical parameter, the capillary refill time, to guide resuscitation, to guide fluid repletion. And since one of the inspiring points of our project is the machine learning and the artificial intelligence, we were quite optimistic in receiving this evidence from JAMA Network Open that the findings of those authors suggest that an individualized treatment strategy based on machine learning can guide the use of capillary refill time to use corticosteroids instead of fluids. If we move to infection screening, infection identification, microbiological identification, the available guidelines also stratify patients depending on the blood culture or test that are able to sketch the infectious agent. Nevertheless, when shock is present, we must start with an antibiotic. But when shock is absent, and this is a novelty of the current guidelines, we can wait, because we can rapidly assess the infectious versus non-infectious cause and then start in a guided way and a cheaper way and most efficient way the antibiotic therapy. But if this is true, we have also to acknowledge that even if we are really good, and I have to be honest, we are not, and we apply our first bundle, the point 2, so obtaining cultures, is crucial. What is happening today? It takes from 1 to 5 days. This is totally unacceptable. So there are novelties that can be implemented. For instance, known culture-based methods to identify microbial pathogens. And in this regard, nucleic acid amplification targets, so narrowed or broad spectrum, and the fascinating agnostic inspired our Pathfinder product. In which terms? Well, to schematize and sketch it and to make the long story short, nowadays we are here. We take blood culture, we wait 1 to 5 days, I have to admit often 5 days, and then we can do identification or gram stain or even MALDI-12 in order to confirm the antibiogram. With the new tools, we might extract DNA, do multiplex or 16S rDNA or circulating free DNA next generation sequencing, and then receive the genus and species identification in 1 hour or 2 hours or maximum 1 day. This, I think, would be really, really remarkable.
Of course, there are advantages. The direct detection of pathogens and the high sensitivity and specificity, the detection of fastidious or rare organisms, but also disadvantages are there. For instance, limitations are constituted by the fact that we can have interference, we can have limits of detection, we can be sensitive to contamination. For instance, a small pilot study at our institutes discovered that Strenotrophomonas is in the environment, and sometimes we can detect Strenotropho, but it's not the causal agent. And moreover, we have to really guide from a clinical standpoint what we are asking to which test, so increase the pre-test probability. Otherwise, we will pick up nonsense data. So, limitation. What is really novel, in my opinion, is this tool, the so-called T2 magnetic resonance tool, that is using an imaging technology to sketch with target DNA in pathogens that are directly found in the whole blood and then lysed, amplified in terms of DNA, and then the amplicon-induced aggregation supermagnetic particles can be detected in nanodrops. In this way, we can detect the clustering chains of the environment in the water molecules that is a mirror of the species that is infecting our patients. For instance, a Candida species. For instance, another fungi. This is doable, even if has still advantages and disadvantages. Like the next generation sequencing, looking for cell-free DNA, has advantages and disadvantages. I would call those techniques a kind of a real needle in haystack. Because, of course, now it's validated. Here is one of the temple of the next generation sequencing. But with next generation sequencing, this takes too long. This is not doable in a small hospital like we mentioned. That's why, even if the company are available for off-the-shelf techniques in these regards, for instance, this CLIA-CAP lab is providing on a relatively small and cheap cost a tool that is able to sketch this phenotype, I would suggest, and we would suggest, to optimize those tools just for exceptional cases. For instance, central nervous system infections that are related to organisms that are not easy to be found, or biopsy-proven invasive fungal infection with simultaneous CNAs localization, and so we can use blood, or septic shock due to fastidious organisms. So exceptions, not routine. To come to the end of my small talk, I would say that gene expression, of course, is something really fascinating for us, but this has been tried, and unfortunately, if we consider arrays and we ask ourselves, can gene expression change the profiling of those patients? The answer is sober. Is sober in which terms? We have a huge heterogeneity. The speakers before me already pointed out. We have data overfitting. So actually, we cannot confirm in several additional validation cohorts the results obtained from arrays. And we need validation and analytic platforms that makes this tool not really doable. So, in order to integrate all those evidence, positive, but I would say also negative evidence, we asked ourselves at the end of the Pathfinder Inspiring project if host responses can be integrated with microbial detection to inform clinical diagnosis. Because I think that the integration of host and microbes is one of the answers. Because if we want to improve the diagnosis, we have to face a fact that nowadays we are looking either at the host or at the microbes. There are tools and platforms like the RNA-seq and DNA-seq that can be really useful in this regard because they can integrate. For instance, here I just reported an example that is published in PNAS in 2018 where the author were able to detect the lower tract infection of the lungs, so pneumonia and sepsis, just based on the high variability of RNA sequencing profile. So they were able to distinguish non-infectious versus infectious pneumonias and SIRS based on the RNA-seq profiling. Why is this a paradigm shift? In my opinion, it's a paradigm shift because using an integrating approach of microbial metagenomics, microbial metatranscriptomics, and host transcriptomics, we can deal with the fact that nowadays we are diagnosing a 21st century disease, sepsis, with tools that were back in 19,000 and with therapy that were from 20,000. So we need a paradigm shift in this regard, and we can do this because if we integrate these real-time microbial metagenomics with real-time microbial metatranscriptomics, of course on small cohort, on discovery cohort, we might generalize the rapid quantification and the rapid cytokine metabolite quantification of the patient in a larger cohort, just based on prediction performed in silico. And this will empower a kind of tool that is really giving us a point of care, exhaled breath analysis or another off-the-shelf tools that is predicted in advance in the discovery cohort. So, in my opinion, personalized medicine is a new hope, but it's a new hope because today we are here. We stratify medicine just randomly. We pick up a patient and we assume that is the same patient that I saw last week, but actually it's not the case. Because drug 1 can be integrated with genomics, transcriptomics, and proteomics of subject 1. Drug 2 can be integrated based on a prediction of metagenomic and pharmacogenetics of patient 3, and these can help to guide clinical trial, as Mikkel told us, that are much more doable, cheaper, and hopefully even more predictable. I would conclude with 3 take-homes. Screening and initial resuscitation is actually something that already changed. We could say that SIRS news is much better in predicting the presence or not of sepsis based on a clinical observation. We need to improve the hemodynamics screening in order to guide our therapy, and we definitely need an implementation in the infectious agent causal identification, because right now we are at the ancestry. With this, I would like to thank all of you for the kind attention, and I would really like to thank all the groups that supported us in this challenging but really amazing project that helps us to help the patient who volunteered, providing us samples and providing us the hope to cope with sepsis. Thanks.
Thank you very much, Antonio, for your excellent, interesting, I would say, really, makes a lot of hope in this kind of problem. So, congratulations, very clear, and I think that it's perfectly, I would say, consistent with the main goals of sepsis and antisepsis. So, thank you very much. Just to tell you one thing before the questions, aquaporin 9 has been reported to increase. There is work done by flow cytometry in humans during the SIRS, so maybe that would be important, while considering the algorithm of artificial intelligence. Thank you. So, questions from the audience?
Yes,
Professor Bilancia.
A question on screening. You made a very important observation that there are a lot of guidelines and that they are not correctly applied. And this calls for a question. Some early warning detection algorithms that I presented before, such CR2s, are very efficient in early recognition of patients. But I am not optimistic that they can be applied in clinical routine. Do you think that the real challenge, the real unmet need is to discover new, simple scores that have higher sensitivity and specificity than QSO for SIRS, but they're as simple Est-ce que tu penses ça?
So, challenging question, and thanks for the questions. Challenging answer. For a lack of time, I did not show, but there are at least two huge registries. One we were discussing together yesterday. In Sweden, they have a huge registry of more than 700,000 patients, and they realized that people who researched for sepsis were not really stupid, because unfortunately we need scores. That's for sure. The problem is the applicability of those. The physician at a certain point do not trust those scores, and they guide based on clinical judgment. So my simple answer would be, we will always need a clinical judgment, but a physician that can understand also those scores and integrate in an intelligible and trustworthy way those tools might be an alternative to just go blind. I don't know if I partially answered.
Yes, you mean that as a clinician you are not confident on the applicability of too much complex algorithm in the clinical practice.
That's why I mentioned
there
the capillary
research, but what is the real applicability?
That's why I pointed out the capillary refill time. This is really basic, but we can do on the bedside. Also a nurse or a 911 operator can do this and discriminate that that patient will evolve rapidly. Okay?
Thank you.
Any other question?
Considering the patient-centered prediction of the pathogen, how far do you think you can push that? Because as I understand from my previous discussions with you, the really important thing would be to detect antibiotic resistances. So I think for discriminating an infectious from a non-infectious pathology in pneumonia, for example, I can easily imagine that this will work, but for discriminating the multi-resistant bacterium from another that is completely unharmful, I see that as a hard thing to do.
I mean, I have no answer for that. I assume that you are right. It's unlikely that we will succeed to fight against multi-drug resistance bacteria just based on those tools. That's why I did not mention multi-drug resistant bacteria. But again, not to be a bit romantic, but in Sweden, based on the implementation of awareness of sepsis, the incidence of multi-drug resistance bacteria was in 2018, I guess, 10%, and now it's much less than 2%. In Italy, in Greece, and France, and Germany, we are not doing really good. In my opinion, this is a problem of prevention and awareness. So I would fight multi-drug resistance bacteria with good habits, no prescription of useless antibiotics, awareness of the resistant cluster, and knowledge of our own epidemiology. For instance, the epidemiology in our clinic can be different from the epidemiology in your clinic.
Thank you.
Okay.
No, actually, it was not a question, it was a comment, but I was already anticipated because I was trying to ask what happens when you don't have drugs able to fight bacteria that are under antibiotic resistance. So I know that this is a real challenge in our world, and underlines the importance to find new targets and new drugs, hopefully, to combine with the mechanisms that we have for the currently available antibiotics. But this is a second point that comes later.
Maybe just 1 minute to try to answer this question, because this is a really unmet need. There's the European Society for Research in Sepsis, and they performed these. This is a backup slide that I took from the registry, because you can see here that Croatia was the most contributing country in providing information. Why? Because they have no money to fight against antibiotic resistance strain. So they were motivated to prevent. So I think the answer is to register the data, know exactly the epidemiology of our bacteria, and then to fight against the conditions that are the predisposing factor to develop antibiotic resistance. Otherwise, I think we cannot make. Yes.
So again, thank you very much, Antonio, for your interesting presentation. So next speaker, Professor Corrado Giuridici, who is an informatician from the Department of Informatics here, and just in front of us. Actually on the left side a little bit, but anyway, in front of us, and with Professor Donato Malerba and the colleague at the University of Lund. They are the AI people in the Antisepsis project. Yeah. So we thank Corrado and Giuridici, Professor Malerba for bringing in this important expertise. So, and really by having something as possible, as most complete possible. So, and in terms of artificial intelligence, because we already submitted the first time the project, and the reviewer said you have to strengthen the artificial intelligence part, and so that's why we are honored to have this important expertise in the new version of the project.
Our honor too.
Thank you very much.
Thanks again for these nice words. Okay, good morning to everybody. In this presentation, I would like to illustrate you what a computer scientist in artificial intelligence can see as interesting, can see as worthwhile to be investigated in the clinical, biological, biomedical research in sepsis. I will do this in general terms, so I hope to not be boring for who is not familiar. Okay. No, it's okay.
Okay.
Okay. Let me introduce some basic notions. Artificial intelligence is a branch of computer science which aims to emulate, which aims to imitating the human behavior, human intelligence, human understanding, even human feeling in machines by exploiting the computational power of the machines, by exploiting the computation machine speed. So this is done in several ways. One way is exploiting the human reasoning. One reasoning type is the induction. We have the inductive-based reasoning. It's a sort of cognitive mechanism that humans use. What we have in this kind of reasoning, we have specific observations of a domain, for instance, of a patient, specific instances. We say that of a phenomenon to derive a general model, able to describe, able to cover, able to I can say generalize those instances. The opposite way is the deduction-based reasoning. Anyway, the AI in general terms aims to replicate the way to think, the way to reason of the humans. The induction actually is the same cognitive mechanism that we have when the kids learn new abilities. What happens? They see, for instance, pictures of houses, they see pictures, houses which have been drawn by other people, so they conceptualize, they realize that they need to draw a roof, walls, windows, doors, window, and so on. That's it, their model of houses, and they will exploit that model to draw new houses. The inductive based reasoning is the basis of a branch of artificial intelligence, machine learning. In machine learning, we have algorithms, tools, able to learn the same capabilities or capabilities close to those of the humans. The capabilities are able to distinguish, to recognize, to summarize, to suppose. This is done by training models from data instances, from specific observation of the domain of our patient, what we call as past experience. A branch of machine learning is predictive machine learning. The speakers before me mentions prediction. Wonderful. We have predictive machine learnings where the aims are to build models able to infer new information, able to suppose information which is unknown at the time of model learning, at the time of clinical analysis, for instance. Just to give you a concrete illustration of predictive machine learning, we can imagine that we have data instances, MRI scans, where the clinicians said if there is or not a pathology. So those data are prepared by exploiting clinical experience. This is the reason we say that data instances are past experience. So we can use those data to feed a predictive algorithm. It returns a predictive model which can be used to suppose, to inference the presence or absence of the pathology on new MRI scans which have been not seen by clinicians. For this reason, predictive machine learning can be helpful in the clinicians' activities, for instance, in this case, in diagnosis. So, to come back to the topic of this workshop, we truly believe that machine learning can be a useful tool not only to support prognosis, which generally tell us that we need provisional information, but I truly think that predictive machine learning can be used also for early detection and diagnosis. Just to give you some example, we can imagine that could be useful, helpful to do prediction of the evolution of vital signs, or to do forecasting of the sepsis onset, or prediction on the probability increase of death. Some case studies. Anyway, to accomplish this high-level purpose, we have to investigate some specific issue of this stuff. We identified three specific issues, three specific challenges concerning data and the design of the algorithms. These are heterogeneity. Michael Antonio mentioned heterogeneity. Heterogeneity, scarcity, and comprehensibility. Heterogeneity, what we have, what is the problem? The paradigm of personalized medicine this project proposal aims to carry on suggests us to see the patient under different perspectives, under different domain, because they can give us several degrees of information. So, for instance, biological data is a perspective, clinical data perspective, pharmaceutical data another perspective, and so on. That is what the personalized medicine sees as a holistic approach in monitoring, investigating a patient. So what is the problem? Actually, the traditional approaches put all this perspective in one algorithm, disregarding the differences between the different perspectives, disregarding the fact that one perspective could be crucial in one prediction, that one perspective could be determinant more than other perspective in the task of prognosis or diagnosis. So what is the point? The point is to emphasize, to exploit this multiplicity of perspective, multiplicity of dimension. What we can do, we can exploit the multi-view machine learning, multi-views, multi-dimensions. So the step is quite straight. In multi-view, actually we have a predictive algorithm, a predictive model for each view. In this way, a predictive model is able to better capture the characteristic of that perspective. The nice thing of this approach, this computational solution, is that these predictive models are learned together, are jointly built, and during this process they help each other in order to obtain more accurate predictions. The first point. The second point is scarcity. Scarcity does not refer to the quality of research, but concerns the data. What happens before? Despite to the biological studies, in clinical studies, when we monitor patients, actually we could not have so many data. We could have a low availability of data, and this happens for several reasons. For instance, data privacy or data protection. You have seen in a talk before this. Data privacy and data protection can lead to incomplete data. Another reason is ethics and regulation. You know better than me that we cannot do blood tests, blood samples on the same patient day by day, week by week. It's not possible. And this leads to have data which are not representative, data which are not significant to learn and accurate predictors which can be helpful for our purposes. That is the problem. One solution is to produce new data synthetically, to produce artificial data. How? We can explore the recent generation of algorithms, namely generative neural networks. Basically, they create new data from the real data we have. Actually, there is not a creative generation of data. It's the same principle, the same idea of the fake images you can find on internet. Fake images are synthetic data, synthetic images which are built, created from real data, where these original data have been a bit perturbated, a bit modified. Of course, there is some complexity behind, but the point is that the new data, the synthetic data, keep the same statistical properties, the same characteristics of the real data. So we can see actually that we have similar data in this new kind of data actually. The third point is comprehensibility. Why comprehensibility? Typically clinicians, unfortunately, but could be, they do not consider, do not account for what AI, artificial intelligence algorithm, artificial intelligence output, artificial intelligence predictions says. Because clearly, because they don't understand how an AI algorithm can work, how an AI prediction has been obtained. So the problem is to make these software tools, these software suggestions, transparent for who is not familiar with computer science. What is the solution? The solution is using another recent generation of technologies, which is explainability, explainable artificial intelligence, which allow us to clarify to the clinicians the causal relationship between the prediction and the input. Namely, with these tools we can explain, we can clarify, we can say to clinicians why the algorithm suggests a recommendation, suggests a prediction by starting point the current condition of the patient in terms of clinical data, biological data, and so on, so on, so on. For instance, in this case, it's just an example to say that these tools, typically they use graphics and visualization, allow us to say that in a specific prediction, this is the ranking of importance used from the algorithm to say that prediction. Moreover, this kind of technologies allow us to clarify what happens for that algorithm with respect to a specific patient, which is another thing, for instance, happening for the same algorithm but for another patient. That was the first point. Okay, thank you for your attention. That was my contribution. Thanks.
Thank you, Corrado. Thank you very much for your presentation. Questions from engineer.
Hi, Corrado. Just an important point. You have shown the importance of synthetic data because in a lot of applications we have no data. And you know some technique about this approach for synthetic data. There are works in literature about this topic because this is my research point.
Fortunately, the domain is very difficult. You know, medical data, biological data are very sensitive and so on, so on, so on. Yes, fortunately, in the literature we can find recent application of artificial neural networks which are able to generate data, linear data, for instance, sequences or spatial data, for instance, images and so on. The point in this case is you have to consider compliance, regulation, and so on. And there is a distrust of clinicians when you create new data. because new data, real data, synthetic data, that do not correspond to a real patient. This is another real challenge. Okay.
Thank you. Any other question from the audience? I have just a small question. Of course, there is already quite a lot of work using artificial intelligence in sepsis. What is your idea about how much is left in terms of improvement in this context?
The first is this one, exploiting the heterogeneity. That is my opinion, and I think the same opinion of Antonio. Considering the several information sources, by considering the different characteristics and properties of this kind of heterogeneity.
Yes. Of course, the more parameters we have available that are, of course, that we can trust, the better will be the algorithm. You know, the more trustable will be the algorithm.
Well, the
it's
point
also a
is,
matter of number of, you know, parameters that you can trust.
The point is not the number of the quantity of the parameters, but the several views you are seeing the patient. Okay? And the point is that you need specific predictor, specific artificial intelligence to handle that kind of data. Actually, we have a sort of funnels, containers, which process all of this stuff and without considering the differences between these different kind of data.
Okay. Again, thank you very much, Corrado.
Okay, thanks.
Thank you. So we can go to the third session. I would like just to say that Professor Alberto Fornasari is sorry for not being able to come because he had a last-minute institutional commitment. So we will have a great expert, Dr. Silvio Bacca, who will share the last session. Please.
Thanks a lot. Not an expert, engaged citizen host. This is the last but not least session. I'm under pleasure to present engineer Alessandro Massaro. He is a professor at Lund University in physics and engineer. Also is a research and development chief of Lund Enterprise. And also he was a researcher at National Council Research and an Institute of Technology, Italian Institute of Technology, the real most important institute of research in Italy. And I think it is one of the best experts in intelligent artificial in the rest of Bari and also so take your time.
Thank you very much for your presentation. Thank you very much also for the invitation of Calamita and of Professor Bacca. And I'll show just another view about technology in telemedicine to see the starting point and we are arrived. And I show some telemedicine architecture and processes. Then I discuss about in artificial intelligence field prediction about diabetes, and I show a Lund Enterprise study in telemedicine project that we have finished in this stage in the last section of the project, ministerial project, and I'll show a research topic that we are carrying out with Professor Bacca and is in a continuous way about the application of artificial intelligence in the dermatopathology. And I'll show some possibility and opportunity to merge our action with the Loom University and the School of Loom about projects in medicine and telemedicine. The many architecture are in my book, and this is a main scheme because in this book you can find also all the possible architecture in telemedicine, and I show just one in which we have a control room and the interaction between the different actors such as doctors, specialists, system administrations, patients using wearable device and laboratories. There are a lot of actors that could contribute in a platform, and different functions can be therapeutic campaigns, auditory campaigns, diagnostic, prognostic, assistance path, psychotherapy, rehabilitation, visit planning, and so on. This is a VPLN scheme of approaches because in Loom University we are studying also the process management, in engineering management, in healthcare, and this is a case of interaction between in a care path process, merging actions of and process of cardiologists, denticians, and psychologists, and in this case the contribution in precision medicine is in therapy and in feedback with feedback control with the clinical tests. And this is a scheme that is an engineering scheme useful for the implementation of process in healthcare environment. This is the first project that we worked with Professor Vaca in InnoLabs Apulia project because this is an important project because it introduced a telemedicine platform and also the interactions between doctors, patients, and laboratories. This project provided different works. This is one because we also merged the clinical algorithm with artificial intelligence one, and we have defined the different thresholds and prediction of parameters. And this is just a sketch about prediction of parameters. Another telemedicine architecture has been shown in this ministerial project of Smart District 4.0 involving Google as partner in partnership of Google, and together with Loom Enterprise, which is a spin-off from the Loom University and the Human University. This is the main scheme of this project because the goal is to control the driver diabetes effect in drivers and involved clothes industry, textile company, and electronic manufacturing of biomedical device for wearable sensors monitoring these parameters, diabetes indicator. This is our design of the platform. We find modeling language that we have implemented in the final prototype of the telemedicine platform, and the results also have been published in the paper. We used, in this case, the long short-term memory artificial neural network to predict diabetes parameters. The background in the long short-term memory is we proposed it in the literature, and we applied our knowledge to the project in order to have new results. This is the final telemedicine platform implemented in Google dashboards, and also here you can see the prediction between, using also artificial neural network algorithm about the glycemic index prediction. This is another work that show the self-learning of peak of diabetes, and also applying the algorithm for different data sets. This is an important point about scarcity that I've discussed before, Professor Lish, because it's an important aspect, because we have usually we do not have enough data to process, and we do not have enough data for the training of the model of the artificial intelligence algorithm. We have implemented an approach that provides synthetic data, augmented data, to predict with low error the results. Then we move with another topic that we are carrying out with Professor Vacca, and in dermatology. We have implemented a powerful algorithm based on supervised fast random forest algorithm to discriminate anomaly in image containing cells in dermatology. This is the algorithm based on classification of clusters of the image, defining the group of pixels, classifying anomalous charts. In this case, it's possible to define probability maps, define cluster of pixels of dangerous area that are classified as alerting area. The algorithm has been implemented in a platform extracting information in the pixel intensity as probability to have anomalous cells, and by also quantifying as with an indicator the percentage of this area in the whole image. These are published results in the paper previously mentioned, by showing also probabilistic maps and the precision of the algorithm that has been applied with a very good performance. The imaging is before classified in different classes, the classes that we found in the final classified image by the supervised algorithm, fast random forest algorithm. We have then implemented and developed the platform by extracting directly the number of pixels in the whole image, pixel highlighted red, and by analyzing the different probabilistic image, extracting information in the image. Yes, then we are working also in a new topic in engineering management, because we started the collaboration with Professor Vacca for COVID management process to define how technology can improve the management of the processes in pandemic situation, and now we are moving on some other application by applying lean approach and process mining by collaborating with the School of Management of the York University. The LUM has an important lab, the Lean Lab, in which work Professor Angelo Rosa, and is a certified lab in healthcare process. This is a slide just showing different projects in the whole Italy, and also has been developed a project with China healthcare systems. And here are all the institutions collaborating with the LUM. This is one slide, and this is another one. Also, LUM works also in research about this application field, and there is a continuous work in this field. Particular thanks to Enterprise, where I am Research and Development Chief, here is also my director, and the acknowledgement is also to to whole Loom ecosystem, Loom Enterprise, Loom University and School of Management, and also for children that we are working also today in continuous way forever. This is. Thank you very much for your attention.
It seems that I have some problem with microphone. Normally I hate microphone, but today I made live, so I. If there are some questions.
Which are, in your opinion, the challenges in applying telemedicine in sepsis?
In sepsis, I think that the first approach is to think the two models to apply. This is the first step. I think that is very, very important. This not only the algorithm, but also the database, the process to select and what you would like to observe. This is a first important point that is the first step to the analysis. I think it is very, very important.
So, if there are other questions, why not I would invite engineer Giuseppe Modugno. Engineer is the chief officer of MTM. MTM is a small enterprise, IT and software house with a long experience and background in IT domain. Engineer Giuseppe Modugno is a mechanical engineer, but he also has a great experience in IT domain. With MTM, they have software, original software and solutions for public and private organizations. Please.
Thanks, Silvio. Good morning, everyone. Okay, I don't want to use a presentation, but I prefer to use directly the application. This application could be used by doctor, by patients, and by nurse. Technically, this application is used. I sit because I have the two hands. Okay. Okay. Perfect. Perfect. No, okay. I have fixed my mobile here. This application is used, as I said, by the patients, the nurse, and the doctor. The application, what is its target? Is to help the patients to be connected with the doctor or with the nurse when he's at home. How it play in this? He play in this in easy way. All you are seeing, it is possible to visualize on the mobile or on the smart glass. What is different with the mobile? You have one hand that put the mobile. With the smart glass, you have two hands free. I can scan with the, what you are seeing. That is the same thing I'm seeing on my phone. What I can do? I can scan a QR code, and in this situation, if in my home there is a IoT instrumentation that communicate with a base, with our application, we see the cardiac frequency, the oxygen, and so on. This sensor, for sure, it could be personalized. So this is only an example. If there is something that goes wrong, an immediately message and an email could send to parent or doctor. If there is something out of the thread, the color of the white data could begin red. So, as continue, it is possible to see. Okay, lo riposiziono. Okay, I can see I'm the patient. This is the example. I can see if there is something. If I don't remember how to use a pressure instrumentation and so on, I could push tutorial, and I see a simple video that the nurse or the doctor could put inside of the application that explain me how to make the pressure, how to do acquire the pressure or other information. It is possible to see my clinic field, or the nurse or the doctor could send me a documentation, for example, a module for the intervention, and I have inside of my application all the documentation needed for all my care. It is possible to see photo or video, the image or video that my doctor could send me. For example, the pills that I have to buy, for example. But if I have any kind, and a particular, the 6 button in the upper side of the application play to solve the problem without the intervention of an external of the doctor. But if I have any question or problem and I need an external help, I can I can chat with the doctor, with the nurse, or I can ask a remote assistance. And in this case, now, from our office, my collaborator make a remote assistance to us with augmented reality and with WebRTC. 1 second, I hope that, okay. Okay. 1 second. Okay. Good morning, Piero. Okay, Piero, do you hear us? Good morning. Okay, you don't earn, but I earn Piero in. Okay, Piero, try again. Good morning. No, okay, it's not possible to ask in the microphone. So, a little imagination. Imagine that Piero is our. No. Okay. Piero is my doctor, and I ask Piero to help me to solve a problem. I'm not able to open the pressure instrumentation and something like this. Please, Piero, could make an augmented reality pointer that help me where I have to put my hand, where I have to solve the problem. I hope you see the red pointer. Yes, okay. And my doctor or my nurse are helping me to solve this kind of problem. Please, it is possible, Piero, a whiteboard image that I don't know how to solve the problem, and I need other kind of support. Piero, my doctor, could create a whiteboard, make a screen of what I'm seeing, and Piero and me, we can work together, we can write, or Piero have indicated me this point, I can react with a simple and easy way. I put the button, okay, I have done with a single finger, and I have the 2 hands occupied here. It is possible to write, it is possible to make number and add and so. No, sta tranquillo, ce la faccio. It is possible to share the video. For example, Pietro could make a sharing information. For example, doctor, I don't know where I have to put my name and surname in the documentation. Piero, open the documentation and indicate me where I have to put. It is possible to make Zoom and so on. This is a collaboration between the doctor and the patient. It is possible to send file and so on. Please, Piero, could send us this file? Okay, I earn Piero. Okay, are you sending it? No. Okay. This application, now Piero is sending us the documentation, and the documentation is on cloud, is putting inside. Okay, this is document, I download, okay, I open with Drive, okay. The documentation we are working before. So this application, it's easy, is easy. It is possible to put in the instrumentation, that the tools that all patients have, the mobile. It's easy. It could put in connection. I say hello to Piero. That is the time for lunch. Thanks a lot, Piero.
Thank you.
You are welcome. So this application put in connection patients and nurse, patients and doctor for remote assistance through augmented reality and WebRTC. Thanks for your attention.
Very interesting presentation, Engineer Olunio. Are there some questions for him?
Is this system useful to send documents or to send X-ray?
For sure, this application have what in technical field we say API. API is like a electrical socket where we can connect it with other platform to send documentation and so on. The platform we have seen before with the Loom, it is possible to integrate our application in that platform because it is developed with a .NET technology with the API connection, so it is possible to connect it with the other software without problem.
Just a general question. How this system right now is accepted? Of course, you know, that is something that helps a lot by the health system. So legally, when I say legally, I say when some insurances are involved, this kind of interaction, how it is taken by the insurances and health care system, the public one also, in addition to the private one.
Okay, about the data, for example, with when we have make the remote assistance, we don't have information protected because we use it a peer-to-peer technology. So this means that we connect the physician with the patient directly with the instrumentation. This means that we don't acquire any other information of a connection and stop. About the other information, radiography and so on, that, as I said from the colleague before, there is a GDPR and we are compliant with that. About the perception of the instrumentation, I think there are a lot of work to do because this solution is more accepted in the industrial field. our application is used in the Magna, Acquedotto Pugliese, A2A, Lottomatica, FPZ, big company that are using it for the remote assistance for the maintenance of the equipment. If you can imagine that the maintenance of instrumentation is similar of the maintenance of the people. We have a lot of work to do in the healthcare, because in this moment it is not being accepted, but I think there is only a question of time. Then Italian government is investing. There's a lot of investment in this solution, with the platform, with the final kilometer. This application is a vertical, is a final kilometer. So my opinion is, my point of view is, it's only a question of time.
So any European country where they are, in advance compared to us.
But the North European are advanced of us. Yes. They are using, they always using this kind of solution. I have read there is Denmark, Netherlands are using this solution.
So they also have the right laws, the legislation.
Yes.
Which is okay. Which I
Absolutely.
with that. Thank you very much.
You are welcome.
Thanks a lot. I think we are going to close the area and the microphone to
Thank you, Silvio. Andrew, please. So, thank you to you, Silvio. Thank you to you. Thank you to Dr. Modugno. Thank you to all of the speakers. And thank you also for the people being in remote. So it was nice for us also to know that people were on the other side. And I think the presentation we had today, they really agree very well with things that might be in remote. So this is a great occasion to show that we can do plenty of things in remote, and things that work, things that are much, much less expensive than things done physically. So it's a good thing, and I thank you everybody. I change to Professor Angelo Vacca, who is the person that gives us the inspiration to do this.
Yes, thanks a lot everybody for this meeting and for coming here, especially Michael for coming in Bari. And I think that new medicine is now, with these projects, new medicine should have collaboration with several professionists, especially IT researchers, psychologists, biostatistical persons, and with beneficial activity to patients and also to spare money in the new health and the new approach to diagnosis and therapy of patients. Thank you very much for coming here.
Thank you to Ambita News for this great occasion. We had, to disseminate, to divulgate this our project, and also it's a way to tell people that we can go very far by using this strategic and I would say a very effective tools. Thank you, Ambita News. Thank you, Dr. Alessandra Rufino. And thank you for also to the technicians who did a great job. And thank you to the audience. Thank you very much.
Viva il Cito!
And thank you to the cheater, of course.